Well, welcome everybody, and thank you for joining us today for this joint webinar given by Fabric Genomics and XIFIN entitled Achieving Success with Next Generation Sequencing, AI-Driven Scale, and a Strong Reimbursement Strategy. My name is Jeanette McCarthy, and I'll be the moderator for this webinar today. Next slide. Today, we'll hear from three speakers: Connor Ward, Vice President of Sales at XIFIN, Clarissa Blattner, Senior Director of Molecular Diagnostics Support Services at XIFIN, and Charlene Sonrigby, Chief Business Officer at Fabric Genomics. Following our speakers, we'll have a Q&A session. So today's webinar will focus on two important areas relevant to labs who are launching next-generation sequencing-based tests. The first is the use of AI to enable labs to scale, and the second is the pathway to reimbursement. In addition, we'll talk about some of the practical considerations for implementing next-gen sequencing, including selecting the right panels, automating and scaling next-gen sequence testing through integrated clinical workflows, including LIMS systems, and clinical services for labs launching or scaling genomic tests. So without further delay, I'm going to turn it over to Connor and we'll get started.
Thanks, Jeanette. As Jeanette mentioned, my name is Connor Ward and I'm a VP of Sales at XIFIN. I thought that I would start things out today by providing a macro overview of the NGS market. According to Fortune Business Insights, the global NGS market was $6.3 billion in 2018 and is expected to reach around $31.4 billion by 2026. That's a compound annual growth rate of 22.2%. If we look at the NGS market by type, we see that approximately 2/3 is product, instruments, software, and consumables, while the remaining third is next-gen services, which are expected to grow at a higher CAGR during this period. The need for novel platforms regarding advancements in bioinformatics and fast DNA data interpretation are paving the way for next-gen sequencing services. Specifically, the key market drivers for this double-digit growth are a huge increase in demand for drug discovery applications requiring NGS technology and diagnostics tests based on sequencing, the rapid pace of new test kits and testing services that are emerging for non-invasive prenatal testing, cancer diagnosis, and others, improving government support through product approval and reimbursement coverage for NGS-based diagnostic tests is providing new market opportunities, and with the expansion of next-gen sequencing, the cost of sequencing has reduced, making genomic testing more accessible. Let's dig into the NGS lab landscape and some of the challenges that we face. We are generating a lot of genomic data. The amount of genomics data being produced on a daily basis is doubling every 7 months. Within the next decade, we are looking at generating between 20 to 40 exabytes a year. For reference, 1 exabyte is equivalent to 366 billion digital pictures with an average file size of 3 megabytes. Or even more plainly, the amount of data my high school-aged daughters consume in 1 month. Beyond managing the vast amount of NGS data, there are still a lot of laboratory challenges, particularly when trying to get paid for NGS services. Today, reimbursement is ever more tied to clinical utility. Still, fewer than 200 CPT codes exist for about 70,000 genetic tests. This means that there is no straightforward way to bill for many tests or for payers to identify what genetic tests were given. And there's a lack of extensive data evaluating the utility and economics of genetic testing. The rapid increase in genomic testing also poses challenges for physicians who are often not well informed on the latest diagnostics. The survey by Stanek et al. Found that 98% of physicians viewed molecular risk assessment playing a key role in determining drug therapies, but yet only 10% felt confident in their understanding of how to best use test results and treatment decisions. Lastly, while this is not unique to NGS labs, there are many systems that ultimately make an integrated and streamlined workflow difficult. From the EHR, sequencing platforms, bioinformatics solutions, we still have our Excel, to the LIS, there are still a lot of data silos within any given lab. Let's take a deeper look at the reimbursement landscape for NGS testing. I'd like to welcome my colleague from XIFIN, Clarissa Blattner.
Thank you, Connor. Good morning to all our attendees on the West Coast and good afternoon for those of you on the East Coast. So I wanted to start off by sharing this slide because it depicts how things have changed with patient care over the years. Providers used to be able to render services needed for their patients based on their best judgment. However, there's now additional strategies involved with the decision-making process, not just for labs, but for all healthcare providers. There also are so many key components you need to factor in before launching NGS testing. So in today's world of diagnostics, there are many changes with lab requirements, technology, payer and test requirements, as well as determining how to balance patient, physician, and payer needs. So I hope the information we share with you today will help you navigate through the challenges many labs face, not only with NGS testing, but across all diagnostic lab specialties, and help develop a strategic business plan to overcome these obstacles. So in 2011, the National Institutes for Health, in collaboration with the National Center for Biotech Information, developed the Genetic Test Registry, which we consider the GTR, to advance public health and research into genetic health and diseases. They actually started recording the data in 2012. This slide actually shows that in 2012, there were about 785 tested conditions, 966 tested genes, with 181 labs registered on their site. Today, there are actually well over 75,000 genetic tests on the market, of which 14% are multi-gene tests and are available for over 11,000 conditions. So in today's market, there are 132 drugs that note genetic markers in their labels to guide treatments, with 42% of drugs in the pipeline that include genetic markers. Precision medicine and genetic testing will continue to grow in importance, use, and spending. However, the current challenges will definitely continue. These limitations include evidence and data, the high cost of testing, variable access to appropriate testing, and most importantly, and what I'll focus on today, is the reimbursement challenges we continue to face. So testing is evolving, and there's been a move from single gene testing, gene panels, whole exome sequencing, to whole genome sequencing. There are also disease-specific testing and inherited genome sequencing versus acquired somatic sequencing. So pulling XIFIN claims data across multiple labs nationwide, we actually compared the number of units of specific CPT codes billed in 2016 as compared to 2019 for a subset of genetic tests. The largest increase that we saw was on the genomic sequencing procedures, the GSPs, and those CPT codes are 81445, 81450, and 81455. The GSP codes represent changes in the irregular arrangements, number, order, and organization within the gene chain specific to the medical condition. This was followed by the BRCA1, BRCA2 full sequence analysis. This displays the exponential growth in genetic testing being performed within the last few years. There are a few key components that should always be considered as part of the strategies for launching NGS testing. Today, I'll briefly highlight the coverage and assignment of codes for billing, which will hopefully result in reimbursement from the payers. Payer coverage defines what services and tests are considered reasonable and necessary. Many payers use what we consider the ACE model process for evaluating genetic tests using 4 main criteria. The A actually stands for analytic validity. The C is for clinical validity. The next C is for the clinical utility, and the E is for the ethical, legal, and social implications. Determining the appropriate billing codes to submit to payers plays an integral part of the journey to reimbursement. Submitting the appropriate ICD-10 diagnosis codes and CPT HCPCS codes provides the payer insight on the patient's clinical history and the reasons for which the test is being performed. Combining both payer coverage and appropriate coding is the pathway to reimbursement for services and tests performed for the diagnosis and treatment of the patient. So payer coverage. In an abstract by the National Comprehensive Cancer Network, NCCN, it was noted that although NGS testing is emerging in oncology practices, there's a lack of formal positive coverage which impacts access and adoption. Payers saw the potential benefits. However, they stated that there were inherent features that didn't meet the medical necessity definition required by coverage. Proactive multidisciplinary efforts to define the direction of NGS development, evidence generation, and incorporation into coverage policies are necessary to realize its promise and provide patient access. There are a few ways to achieve payer coverage for NGS testing. CMS National Coverage Determinations, or NCDs, are made through an evidence-based process with opportunities for public participation. The Medicare Evidence Development and Coverage Advisory Committee was actually established to provide independent guidance and expert advice to Medicare. They often play an integral role in the review and evaluation process for coverage determination. In the absence of the national coverage policy, services and tests may be covered by Medicare administrative contractors, or what we refer to as MACs, as a local coverage determination, or LCD. Many payers have their own processes to determine medical coverage to help justify medical necessity through evidence-based clinical guidelines. Payers also develop utilization management programs as a primary cost containment strategy. Examples of these clinical utilization management guidelines and medical policies may not always align with CMS guidelines. So in March 2018, CMS began covering NGS tests for patients with advanced stage cancers that met specific criteria. In early January of this year, CMS expanded their national coverage determination to include coverage for patients with germline, breast, or ovarian cancers. As part of the revision, CMS also gave regional MACs, or Medicare administrative contractors, discretion over whether to cover certain other indications through their local coverage determinations with the following conditions. It would be for any cancer diagnosis, a clinical indication for germline testing of inherited cancers, a risk factor for germline cancer, and the patient has not been previously tested with the same germline test using NGS for the same germline genetic content. So under the MolDX program, which is actually managed through Palmetto, 2 draft local coverage determinations were proposed in mid-2019. DL38045, NGS for solid tumors, and DL38047, NGS lab developed tests for myeloid malignancies and suspected myeloid malignancies. Stakeholders commented on the proposals during a comment period, and the coverage guidelines are still in progress to further determine the coverage indications, limitations, and medical necessity based on the technical assessment and stakeholder comments. The MolDX program, as mentioned, managed by the Palmetto GBA, requires a technical assessment for all laboratory developed tests, or LDTs, including NGS technology to determine coverage. The assay must demonstrate clinical utility, fulfill the CMS reasonable and necessary criteria, and meet analytical and clinical validity standards. They also follow the ACE model process, which I mentioned earlier. The MolDX program applies the technical assessment requirement not only to new tests seeking coverage, but also to tests that are currently covered to maintain coverage. So correct code assignment can eliminate back-end denials. Therefore, you need to know the appropriate codes to bill and if the payer has a preferred coding approach, because it can differ from payer to payer. So the Tier 1 molecular pathology codes represent gene-specific and genomic procedures. Tier 2 molecular pathology codes are used to report procedures not listed in Tier 1 molecular pathology codes and are arranged by level of technical resources and interpretive work by the physician or other qualified healthcare professional. The GSP, or genomic sequencing procedure codes, are targeted NGS panels for somatic variant detection. And you have your MAAA codes, your multi-analyte assays, which are the procedures using results of multiple assay panels to perform algorithmic analysis. And then there's the PLA code, or proprietary laboratory analysis codes. And those are assigned by AMA to report a given proprietary laboratory service or test. And lastly, the unlisted molecular pathology code of 81479 should only be reported for a unique procedure that is not adequately addressed by any CPT code. So on this slide, we at XIFIN actually compiled claims data across customers that we have nationwide to show the average reimbursement excluding denials for a few NGS tests. This was a blended average for the major payers, both contracted and non-contracted, for Aetna, Anthem, Blue Cross Blue Shield, Humana, and United Healthcare. We also included Medicare allowable rates as a baseline. So as you can see, 81162 and 81163, which represents the BRCA1, BRCA2, had an average reimbursement of $1,100. CPT code 81415 and 81416 for the exome sequence analysis had an average reimbursement of $3,000. CPT code 81432 for hereditary breast genomic sequence analysis had an average reimbursement of $635. 81435 and 81436 for the hereditary colon genomic sequence analysis had an average reimbursement of $400. And lastly, CPT code 81420 and 81507 for NIPT had an average reimbursement of $500. So this next slide includes the average reimbursement for the genomic sequencing codes. Reimbursement averaged around $345 for 81445, $550 for 81450, and about $2,500 for 81455 respectively. So on the previous two slides, I provided claims data on the average reimbursement for NGS codes, which I noted excludes denials. This slide here shows the units billed and the units paid. With that, 61% on average being paid for the 2019 NGS claims. So based on the previous slide, I mentioned that 61% of the units billed were paid, and that leaves about 39% unpaid due to denials received from the payers. The highest volume reason code and remark codes received were due to experimental and investigational, medical necessity, and prior authorization denials. Many other denials were due to payers requesting additional information in the form of medical records, clinical history, and test results, just to name a few. Providers can dispute a payer's decision to deny a claim. Many payers offer rights to appeal a claim or go through a reconsideration or redetermination process. Payers often require an appeal letter or payer proprietary appeal form with medical records. It's imperative that the patient's medical records are well documented by the ordering or treating physician, and it does include the relevant diagnosis information, any previous procedures, treatments, or therapies, relevant test results and pathology reports, clinical history, and family pedigree, and why the test was ordered and how the test results will impact the physician's decision or the management of patient care. So most of our biggest payers, Aetna, Cigna, United Healthcare, and a lot of other major payers have published policies that require prior authorization for molecular genetics and NGS testing. Some payers actually require that the ordering or treating physician needs to initiate that prior authorization. Some payers like Cigna even require genetic counseling prior to the testing. This has definitely placed an administrative burden on physicians who are ordering the tests for their patients because much of the authorization process is still a manual process. This has also resulted in a delay of testing as well as a significant increase in prior authorization denials. So if you look at this slide, across all labs, regardless of lab specialty, there's been an over 300% increase in prior authorization denials. There's 92% increase for labs with proprietary tests and a 52% increase for molecular labs over the last 24 months. Based on XIFIN cross-customer data, if you look at trends across lab specialties, we've seen a 55% increase in prior authorization denials for cardiovascular disease, 73% increase in oncology, and 62% increase in women's health. I also noted that there's been an increase, or actually a 40% abandon rate due to the complex prior authorization approvals process. So many of the major payers are leveraging third-party administrators such as genetic benefit managers and laboratory benefit managers like AIM, Beacon, and Evacor as a cost containment strategy. They are also requiring prior authorization on all molecular and NGS testing. All 5 major payers, which include Aetna, Anthem, Cigna, Humana, and United Healthcare, all have clinical guidelines and or medical policies either requiring specific clinical indications and or specific diagnosis codes that justify medical necessity, or indicate that NGS testing is either experimental and investigational or not medically necessary. Therefore, you want to stay abreast of payer billing requirements and clinical guidelines and medical policies to ensure compliant and required billing practices are being followed accordingly. So based on the current landscape and challenges for NGS reimbursement, it is highly recommended that labs engage with payers, clinicians, and patients. Engage with the payer strategically and have direct contact with a medical director or policy advisor, and educate the payer for access expansion. Leverage key opinion leaders within the industry to support with communications to the payer and provide subject matter expertise on the importance and benefits of your test. Ensure you align with payers on clinical utility and evidence requirements, as well as billing policy and correct coding approach, as it may differ from payer to payer. It is also important to engage with a clinician on medical necessity of the test, ensuring that they order the test with the appropriate clinical indications and document the patient's medical record accordingly, documenting what test was ordered, why it was ordered, and how the results impacted their decision and or patient management and care. Also collaborate with the clinician on prior authorization processes, since many payers require that the prior authorization is initiated by the ordering or treating physician. One strategy that labs often overlook is patient engagement and education and understanding the billing process and possible patient responsibility. A patient can actually be a great asset, resource, and ally to work with the payer to expedite your prior authorization request and appeals process. So this concludes my portion of the presentation. So I'm going to go ahead and give this back to Connor.
Great, thanks, Clarissa. All right, so I'll briefly take the reins back to talk about some of the strategic and tactical considerations for managing an NGS testing program in your lab. My background is in lab systems, and the LIS, or LIMS, is really a key component in the data flow and data management process. At the end of the day, the product of an NGS test is the lab report that the LIS produces and distributes. The primary message and takeaway from this slide is that the LIS is a key component of the NGS lab workflow, but it doesn't function in isolation. For example, any scalable LIS should have an open API that enables connectivity to a system at the point of order, such as an EMR. Within the lab, it should also provide connectivity to instrumentation, bioinformatics systems or services, such as those provided by Fabric Genomics, and of course, on the back end, it should also provide integration with a billing platform in order for the lab to get paid for its services in a timely manner. This is a simplified visual of an overall NGS workflow, starting at the point of order with the physician. The order can come in electronically to the lab's LIS if an interface is in place to an EMR, or can be processed manually at the lab if it arrives on a paper requisition. Even if it arrives on paper, there are batch processing or multi-step accessioning processes that can reduce the bottleneck at the lab that accessioning can often create. Once in the lab, the LIS's role is to streamline workflow using discrete work queues that keep users on task and provide sample progress visibility. I'll talk about some of the in-lab processes in more detail in the next few slides, but I want to highlight the importance of a tight integration with a billing platform, such as XIFIN RPM. As Clarissa mentioned, for NGS in particular, prior authorization or insurance verification may be required for tests in order to get paid. These processes generally occur outside of the LIS, but the LIS plays a key role in transmitting complete and timely data including potentially prior authorization codes to the billing system. In an ideal world, the LIS and RCM platforms will share client-facing portals that support order entry and prior authorization initiation. I mentioned that I had discussed some of the relevant in-lab processes. I've exploded out the black box from the previous slide to provide more detail into the NGS workflow that occurs outside the LIS. Again, this is where our friends at Fabric Genomics can play an important role. At its simplest, the bioinformatics workflow takes a huge amount of raw data, identifies variants relative to a reference human genome, interprets those data, and then reports out any target variants of clinical significance, either in a final PDF report or as discrete data. This final step, which may include a review process prior to sending out the report, is where the handoff from the data analysis platform back to the LIS occurs. I often get asked what the difference is between an LIS and LIMS. My usual tongue-in-cheek answer is one letter, or that LIMS is a four-letter word. In reality, when comparing LIS versus LIMS, the traditional difference that is cited is that LIS are patient-centric with an emphasis on keeping PHI secure, while LIMS tend to be sample-centric. At the end of the day, though, both systems automate workflow, manage samples, and produce test results. What I've seen in the NGS market is that many labs may have used a LIMS for research and wet lab specimen management prior to commercializing their NGS test. The LIMS works well in this capacity, but it needs the front-end client and patient management functions and the back-end reporting and billing systems integrations that an LIS provides. Rather than start over from scratch or build the entire workflow in the LIS, an integrated LIS LIMS platform is a viable alternative. At XIFIN, we refer to this approach as the bookend approach. It relies on seamless data exchange between the two systems, and it preserves the investment made in LIMS while providing the critical commercial functionality of an LIS. While a flexible and scalable LIS should provide end-to-end workflow, the bookend option has worked well for many NGS clients. When we talk about wet lab workflow, we are referring to sample processing steps versus dry lab, which are the data analysis and reporting tasks. The wet lab processing steps may include DNA extraction and quantification, RNA to cDNA conversion, followed by a library preparation step as an example. As discussed in the prior slide, this workflow can be handled in separate LIMS or LIS systems, or if the LIS has a robust wet lab tools, it can be handled in a single end-to-end system. Ultimately, though, you want a system that has the flexibility to configure each unique workflow step, create and manage batches of specimens, and build flexible plate maps and lab-defined procedures that effectively serve as your lab's electronic SOP. The user-defined lab procedures, or SOPs, in the LIS are a way of capturing and documenting all of the wet lab information, including controls, reagents used in a master mix, calculations, and of course an audit of the user completing the work. Historically, and still today in many labs, this work is often managed in Excel spreadsheets. At the end of the day, the report is the product to the client. It's important to work with your vendor to design a report that is both visually appealing and simplifies what can be very complex data to the physician and ultimately the patient. In my opening remarks, I mentioned a stat that found that 98% of physicians viewed molecular risk assessment playing a key role in determining drug therapies, but yet only 10% felt confident in their understanding and how best to use test results and treatment decisions. If critical data is buried deep in a multi-page NGS report, chances are time-strapped physicians aren't going to find it. And lastly, from an operational standpoint, the old saying holds true that if you can't measure it, you can't improve it. So look for your LIS and your RCM systems to provide the insight into your turnaround time, client specimen or documentation issues, client volume stats, and of course, claim submission rejections and appeal success rates. The data is in there. All right, so at this point, I'm going to transition to my colleague, Charlene Son-Rigby, who's the Chief Business Officer at Fabric Genomics.
Great, thanks Connor, and thanks Jeanette. Today, I will discuss key considerations and steps that a clinical lab should be taking in a new or expanding NGS program. I'm going to focus on informatics as well as highlight some aspects of the wet lab. Fabric Genomics brings significant expertise in informatics, and it's also where the majority of a lab's time, effort, and total cost in the NGS workflow might be spent. And this is because next generation sequencing is a big data problem. Due to the amount of data, labs can get bogged down, and it can be in mundane areas like simple data handling and going on to more sophisticated analytics that are required for NGS. And this is especially true contrasted with older molecular testing technologies such as qPCR or fluorescence in situ hybridization. So in my presentation today, we'll cover methods, we'll cover considerations for methods, test design, interpretation, and staffing. I'll also discuss the value of artificial intelligence approaches to scale testing efficiently and effectively. So first, a little bit of background on Fabric Genomics. We've been serving the NGS market since 2009 and working with clinical customers and key partners, developing an advanced and cost-efficient clinical analysis platform. We have a global footprint, and we're primarily focused on high throughput hospital and commercial labs, as well as large-scale sequencing programs. We offer a complete NGS informatics solution from the raw data off the sequencer to the final report, and that's for any NGS assay, germline, as well as oncology. For today, I'm going to focus on smaller hereditary panels, which we find are the best starting points for labs in NGS. I want to note that artificial intelligence and machine learning are fundamental to our platform, and we have a family of algorithms to speed and improve interpretation called Fabric AI. From an IT perspective, our platform is cloud-based and regulatory compliant, and it's very easy to integrate with other systems using our API, including with our valued partner XIFIN's LIS. And we've also introduced value-add clinical services for interpretation and sign-out to support our lab's fast launch and ability to scale quickly. The positive reimbursement trends that Clarissa covered earlier and the dramatic decrease in sequencing costs, as illustrated on this graph on the left, is driving the growth of NGS testing. Labs are finding, however, that NGS is different from traditional molecular techniques such as qPCR or FISH. With NGS, there's a dramatically larger amount of data that labs have to deal with because regardless of the size of your NGS assay, you're covering a lot more territory compared with very targeted traditional molecular techniques. This is great because it means new unparalleled discovery in a single test and the ability to capture biomarkers without prior knowledge. Not only that, you can also capture more complex targets such as small and large scale deletions and duplications. NGS provides the ability to reduce testing burden within the wet lab with greatly enhanced multiplexing. But before capitalizing on these advantages, there are some initial hurdles to overcome. First, the NGS workflow is more lengthy both in the lab as well as in analytics, so it requires some effort to get up and running compared to, for instance, multiplex PCR. More importantly, as I mentioned before, NGS presents a quintessential big data problem, challenging labs to be able to interpret and make sense of this voluminous data in a scaled and cost-effective way. More specifically, labs need to determine how to quickly and accurately identify the pertinent information to analyze and report on. So how to report on novel biomarkers, how to handle incidental findings, and also how to manage and track rapidly evolving clinical and research evidence to support gene and variant analysis. The first decision in a starting or evolving an NGS program is test design, and there are a couple of factors in the decision. First is the size of the panel. Smaller panels are obviously lower cost, both in the wet lab since you can batch more samples in a single sequencing run, but on the informatics side as well. That said, larger panels and even exomes can be very attractive because they'll feature nearly identical fixed costs in setup and maintenance, but offer the potential for a broader testing menu without having to scale lab operations. This includes not only testing more targets, but offering different virtual tests on a single wet lab assay, just by altering the focus on different genes on the informatics side. And I'm going to show you an example of this in the next slide. At a higher level, here are some key considerations as you plan out your NGS infrastructure. First, how reimbursement, as discussed by Clarissa today, is driving your cost requirements. Second, what your lab's performance requirements are in terms of accuracy and other parameters. So larger assays will require more setup, more effort to set up in this regard. Third, what are your lab's goals, both near and long-term, for increasing volumes and expanding your testing menu, as well as for targets for follow-up and reflex testing? Here is an example of how multiple tests can be run on a single wet lab assay. This is an example of a 100+ gene assay, which can be used in multiple virtual tests for hereditary cancer testing. So starting in the middle, a lab can offer a BRCA1 and BRCA2 test, and that's with or without deletions and duplications. The inclusion of these deletions and duplications has implications for reimbursement, as you can see from the CMS numbers below. A lab can also offer a hereditary breast and ovarian cancer panel, or HBOC panel, shown in the light blue, and that includes BRCA1 and BRCA2. The lab might also run a colorectal cancer panel, and that has overlapping genes with the HBOC panel, reinforcing the value of running these multiple tests on the same wet lab backbone. And the lab can also run larger tests like a pan-cancer panel. All of these tests can be run on a single comprehensive panel assay, such as the Illumina TrueSight Hereditary Cancer 113 gene panel. And this enables a lab to offer a broad test menu while running an efficient wet lab process with a single NGS assay. Moving on to the interpretation of variants, it's important to highlight that NGS represents a unique challenge in terms of the scope of information it provides. As a point of reference for a typical small panel, say for hereditary cancer risk, you'll see at least a couple dozen variants that must be interpreted before any reporting can be done. And then this number really grows in tandem with the size of the assay, leading to 100,000 or more variants in an exome. The challenge in panels is all variants must be reviewed for pathogenicity, evaluating them based on multiple lines of evidence. And at the same time, most variants are not pathogenic. So this represents an efficiency problem. The American College for Medical Genetics guidelines provide standardization with 28 criteria that can be answered for every variant identified. There are some complex rules that combine these criteria, so it's not as simple as answering 28 sequential yes/no questions. As noted before, the key pathogenic variants will not often be obvious prior to reviewing clinical evidence, and thus there's a need to assess various sources of evidence simultaneously, including reported phenotype, mutation databases, prior related cases, computational predictors, and research literature. This process can be very manual and time-consuming, and labs need tools to use all this evidence in a way that maximizes their diagnostic yield and minimizes staff time and effort. Labs also need a well-defined, repeatable, and scalable workflow. This is defined by algorithms and SOPs that enforce industry standard interpretation, such as that recommended by ACMG. Finally, while your lab may have access to databases of publicly available variant classifications, the lab will also want a living database of variants that have been seen in its own tests. Reusing these prior classifications is a key way to minimize burden on your team. To ensure NGS testing is not only fast but accurate, it's important to consider the demand that's being placed on the geneticists who will use the system, probably more than anyone else in your lab. A key goal should be minimizing their effort on each case, preventing errors, and delivering consistent reporting. Artificial intelligence and machine learning are not just buzzwords in this context of NGS analysis. There are now established use cases demonstrating the time and effort savings, especially important when considering that informatics, interpretation, and reporting can take up the majority of your NGS testing efforts. This includes automating much of the complex ACMG classification process. Also to consider, there's a lot of latent knowledge out there in disparate public resources, from clinical reports to other more basic research and literature. AI is being applied very successfully now to unlock this latent knowledge, allowing for even greater confidence and diagnostic yield in NGS testing. One thing I'd like to stress here is that these technologies are not typically about replacing staff, but rather about maximizing their efficiency and performance, automating repetitive tasks, providing the right contextual information, and freeing their time to apply their genetics expertise to the difficult cases. So now turning to the Fabric Enterprise platform. Fabric Enterprise is a comprehensive platform driven by a proprietary set of AI technologies called Fabric AI. These technologies accelerate and improve the accuracy of genomic interpretation. Fabric AI technologies span use cases across NGS testing, and these algorithms include Fabric Gem for whole exome and whole genome interpretation, which we just released last month. Fabric AI also includes Fabric ACE for automating classification of variants in hereditary panels. Fabric ACE has been extensively validated over 100,000 variants and enables labs to implement rapid ACMG compliant classification in a scalable, reliable, and repeatable way. With Fabric ACE, the vast majority of variants in NGS panels can be classified instantly, then only requiring a short review by a geneticist instead of the typical 15 to 30 minutes per variant. Here is a high-level view of the NGS analysis and reporting workflow. Fabric starts with the raw data from the sequencer and delivers insights that can be used in clinical management. Our end-to-end platform provides automated AI-enabled workflows. We can start with FASTQ or VCF files and take those files through secondary as well as tertiary analysis, and then generate clinical reports either as formatted PDFs or structured output via our API. And from here, the report moves to the clinician to help manage the patient's clinical care. With this end-to-end workflow, labs can deploy tests for a broad set of medical conditions, including the panel testing we focused on today for cancer risk, as well as for cardiovascular conditions. Fabric Genomics also has a network of clinical services resources for interpretation and sign-out. These include experienced geneticists, many of them ABMGG boarded or board eligible, with broad experience across disease areas. Clinical services can enable labs to launch testing quickly while still ramping up staff, or for long-term needs for interpretation. In closing, Fabric Genomics enables the end-to-end NGS workflow from raw data off the sequencer through to clinical report. With our AI-driven platform for genomic interpretation, we work with key partners to enable labs to easily develop, launch, and scale reimbursable NGS testing. Thanks for your attention. I'll pass the mic back to Jeanette now.
Thanks, Charlene, and thanks, Connor and Clarissa, for your presentations. We're now going to move into the Q&A part of our presentation. We do have some great questions coming in for the speakers. The first question is for XIFIN. Does the lab typically have to rely on the LIS vendor to build new lab workflows and wet lab procedures?
Yeah, I can. This is Connor. I can jump in and take that one. The quick answer is that an LIS or LIMS vendor can provide assistance setting up workflow and lab procedures in the system, but they should do so using a configuration tool set via the user interface versus back-end coding. The end user should then be trained to use the same tools going forward so that they're not reliant on the vendor to create new workflows or make updates or adjustments to existing ones.
Great. Next question also for XIFIN. How do you determine the correct coding for next-gen sequence testing?
Hi, Jeanette. This is Clarissa from XIFIN. I can answer that. So if your test has a PLA code, that actually takes precedence. So that would be your first selection. If your test has an algorithmic analysis, you would actually assign a MAAA code, so your multi-analyte analysis code. For the GSP, the genomic sequencing procedure codes that look for changes in the order, organization, and irregular arrangements within the gene chain specific to the condition, you would report 81445 and 81450 for the panel that evaluates solid organ neoplasm, where 5 to 50 genes are examined. Or if 51 or more genes are examined, that would be reported with the 81455. And these specific codes include the interrogation for sequence variants and copy number variants or rearrangements when performed. So when all of the components of the procedure are not performed, you would report the appropriate tier 1 or tier 2 code instead. And in the event that the service is not identified by any of those particular codes, then you would assign 81479, which is a miscellaneous code.
Okay, great. And as long as we're on 81479, which thank you for reminding me what that code was. The next question asks, I've seen labs bill 81479. Can you bill that code and what is the reimbursement?
Sure. So you should only bill the 81479 code if there's no other CPT code or HCPCS code that actually describes the test performed. Because of the fact that it is a miscellaneous code and it's considered the last resort, payers actually will review this particular code or the claim that has the 81479 on an individual basis. So it may actually take a little longer to adjudicate the claim. As far as reimbursement is concerned, it actually varies because an 81479, as mentioned, is a miscellaneous code. So it could be described for any different type of testing. So whether it's molecular, specific NGS, and so the reimbursement for that can vary dramatically. So we've seen anywhere from, let's just say $250 to $300 in reimbursement, all the way up to, let's just say $5,000 to $7,000 in reimbursement.
That's great. Next question is for Charlene. Does Fabric Genomics interpretation software need FDA approval or clearance for clinical use? This is a really great question. I've had a number of people ask me this in the past. Charlene?
Great, thanks, Jeanette. So the Fabric Enterprise platform is typically deployed by our clinical labs and utilized within their laboratory developed testing process. So that's under CLIA or CAP or similar in other countries.
Okay, great. Next question also for you, Charlene. Do you use variant annotation and any other tools to automatically calculate the ACMG pathogenicity?
Yes, so unfortunately we didn't have time for a demo today, but our ACE algorithm utilizes multiple lines of evidence and annotation to evaluate each variant and generate an initial classification. And so that initial classification then will be reviewed by a variant interpreter or geneticist within the lab to confirm the classification and then determine whether it should be reported out.
Next question also for you, Charlene. Are there issues with AI and machine learning creating a black box that obscures an NGS reporting process?
Yeah, that's a great question, because I think that this is a concern for, since artificial intelligence techniques and approaches are relatively new in this context. This is a concern that we've heard from some potential users. And from a philosophical standpoint, Fabric has always really taken a transparency approach. So the intent and the way that our AI has been implemented is really a support to the end user in terms of being able to make the appropriate decisions as to what should be included within a clinical report. And so as part of that, we present, so with ACE, we're presenting that preliminary classification, and then we are very transparent, providing all of the evidence that was generated to come up with that preliminary classification, so that the end user can then evaluate it and confirm the classification and finish the report. And that's very similar with our other algorithms such as GEM, FAST, and Fever.
Another question for you, Charlene. Does Fabric's platform include CNV or fusion analysis? In other words, what's the capability of the software for handling structural variants?
Oh, great. Yes, so the Fabric Enterprise does support the analysis of CNVs or del dupes or fusions. And what we do is we provide an integrated analysis, so those can be analyzed in the context of small variants, since that can often be important for the overall diagnosis of the patient. And we offer annotation databases that are specific to CNVs, as well as we have for whole genome data, a structural variant viewer as well.
Next question for you, Charlene. If I reanalyze the raw data file, can I augment the original analytical results?
I guess there's two ways that I would interpret this question. So first, if one were to reanalyze a data file, say a year later, to see if there's been any there has been any change in terms of the classifications, that is definitely something that we can support within the Fabric application. So an existing VCF, or if you were to start from a FASTQ, can be annotated again with updated annotations, and then that data can be reported out again. This question also might be talking about reflex testing. And so in the case where you have a virtual panel where, say you started out with a very small set of genes of 10 genes, and then you wanted to reflex out to a 100 gene panel, that's also possible to do in the platform.
Okay, great. Yeah, I think reanalysis is an interesting topic. I'm not sure what your experience is at Fabric with that, but I know, for example, a lot of labs, they don't do it automatically. Is that your understanding? And it needs to be requested by a physician?
Yes, that is what we are seeing in terms of our lab clients. There's tremendous interest in being able to move to something where patients would be able to, that process would be more automated. And from a technology standpoint, that's of high interest to us. But today, or historically, what we've been seeing, it's been more on a physician requested basis.
So we are just out of time. We're at the top of the hour. So if we didn't get to your questions, someone from Fabric or XIFIN will reach out to you directly. You can also reach out to us at info@fabricgenomics.com. And you can also check out our website, fabricgenomics.com, if you want to have any other questions about our technology or view any of our other webinars that we've given. This webinar will be recorded and will be available on demand in a few days. You'll get an email to let you know when it's available. And stay tuned for more webinars to come from Fabric. I want to thank our speakers again and thank all of you for attending today's webinar. Have a great day.